Hello everyone, hope you all are fine. Stay blessed, stay healthy. Here is Dr. Hira Bashir, MPhil Pharmaceutical Chemistry, and we will discuss here staining. What is staining? Basically, it's a process of giving color. As we were discussing about microorganisms, so for microorganism, the staining is basically gram staining. Alright. And staining is basically of three types: simple staining, differential staining, and special staining. Simple staining is of further two types: direct staining, means positive staining, and indirect staining, negative staining. Different type of dyes are used on the basis of which positive and negative word is assigned. For example, if acidic dye is used, then that staining is indirect staining, and if basic dye is used, the staining is positive staining. Then in case of differential staining, there are two different types like gram staining and acid-fast staining. Gram staining stands for gram positive as well as gram negative bacteria. Gram positive and gram negative bacteria. And for acid fast staining, acid fast bacteria are detected. Acid fast bacteria are detected. Special staining includes capsule staining, endospore staining, and flagella staining. Capsule, endospore, and flagella, all three are different parts of the bacterial anatomy. And if we stain these three different parts specifically, then the type of staining differentiated. But if we stain positive bacteria or negative bacteria, the whole bacteria is stained, not anatomical part is stained. The whole bacteria is stained. Then if positive, then gram positive staining. If the secondary dye retained, then gram negative staining for gram negative bacteria. Alright. Then hope you all clear for this simple staining, differential staining, and special staining. What are the basic protocols for staining? What are the basic protocols for staining? The protocols for staining generally involves three sequential stages. Basic three stages are involved. The first one is the preparation of smear. Preparation of smear. Now what is smear? You can see here in the slide. This is a smear. How it is prepared, how it is fixed, we will see here. The first part is the preparation of smear. The other is the fixation of smear. We prepare a smear, then we fix it. Then stain the specimen. This is then stained. This smear is then stained. Now what does smear contain? The smear preparation, it is primary stage, the first stage, which involves mixing of inoculum. As we have studied earlier about inoculum, which is basically the sample of microorganisms which is to be identified, which is needed to be identified. That original sample or inoculum with the drop of sterile water, purified water, distilled water, we take a sterile water. From all different types of water, we select the sterile one, having 100% microbial free nature. Okay. With this drop of sterile water, we mix that inoculum we have on the loop. Alright. Take a loop, put it in the oculum, then take the inoculum and put it on this slide. Put one drop of sterile water until the formation of a thin film over the glass slide, and form a thin film. Very thin film is formed over it. Then fixation of smear. It is a secondary stage. The second stage after preparing smear, it involves heat fixing. We use heat to fix this layer of inoculum on the glass slide. Okay. Secondary stage, heating the glass slide having the smear, and then that smear is fixed on the glass slide. Then thirdly, staining is performed. It is a final and important stage that adds color to the microscope matter. When we see this slide under microscope, to differentiate the bacteria, there is a color which we add during staining. Then staining is the final stage for identification, for examination, for performing biochemical test. This is a smear preparation. We first take a clean glass slide. Alright. Then add a drop of distilled water there and put inoculum here. Mix this loop contain inoculum. This drop of water and inoculum both are mixed properly and a thin film is formed. This is the first step. A thin, clean, dried glass slide. Distilled water drop is put over it. Then with the loop having inoculum mixed with drop of water. Then differential staining, it differentiates between the physical and chemical properties of two different groups of organisms. Depending upon the cell wall characteristics, it can be used, and it makes the use of multiple or more than one stains. It can be categorized into two types. What it is saying? It says that the physical and chemical properties of two different groups of organisms are taken. For example, the groups of organisms may be gram-positive bacteria and gram-negative bacteria. As I told you, gram-positive contain more peptidoglycan in their cell wall and gram-negative contain less peptidoglycan in their cell wall. And gram-positive stained purple and gram-negative stained pink. How they are stained pink or purple? Due to the presence of peptidoglycan in their cell wall. The concentration, the quantity of peptidoglycan in the cell wall. It is further categorized into two types, gram staining and, as I told you, about acid fast staining. We will discuss here about gram staining first. It provides an important tool to differentiate two major groups of bacteria, gram positive and gram negative bacteria. Dr. Christian Joachim Gram introduced this method. As I told you, Gram is the name of microbiologist who introduced this method of staining. Then the complete name is Dr. Hans Christian Joachim Gram. Introduced this method in 1884. It is carried out by the use of differential stain known as Gram stain. Gram stain is used to differentiate these two types of bacteria, gram positive bacteria and gram negative bacteria. Gram positive bacteria and gram negative bacteria. Then gram stain, it differentiates between bacteria into two broad groups. Positive appear violet. You can see here under microscope this type of slide is seen. Visibly you cannot see this, but under microscope it look like this. And gram negative appear red. You can see here these two. This one is pink or red color for gram negative, and this one is violet color for gram positive. You can see the color. This one is for violet and this is violet color, and this is red, or this shade is red, and this one is for both. You can see here slightly reddish color somewhere, and somewhere there are violet color. The gram stain consists of following steps. First of all, we have to prepare the smear. The first step is the preparation of smear and fixation of smear. Then the third step was staining, which we are discussing here. The first step of staining is the use of primary dye, that is crystal violet. Stain both gram negative and gram positive bacteria. We take a stain of crystal violet color. These are gram positive, these are gram negative. You can change the shape. This could be gram negative, this could be gram positive. But in the end, the change will be shown on the basis of color. Both have peptidoglycan, but positive contain more peptidoglycan in cell wall and negative contain less peptidoglycan in cell wall. Stain both gram negative and positive bacteria. This is crystal violet dye. Both are stained, positive and are stained, negative are stained. Then iodine, gram iodine is taken, which fixes this stain in gram positive bacteria. When we apply gram iodine, you can see here in third step gram iodine on this one and this one. Both take this gram iodine and then we wash this with ethanol or acetone. Basically these two are decolorizers. You can also say these are decolorizers. Washes the stain for gram negative bacteria. Then after adding ethanol, the gram positive retain this color, this violet color. After adding gram iodine, this violet color is retained, but gram negative decolorizes. They decolorized and become colorless. The violet color is discharged. Then there is a secondary dye, saffronin, counter stain, which is also called as counter stain. A simple stain is crystal violet dye and counter stain is saffronin. It will restain gram negative bacteria while not interfering with the positive stain in the gram positive bacteria. You can see here both are stained and these are, then the color will be pink for gram negative or red for gram negative, and this crystal violet is retained. Alright, decolorizer removes the stain from gram negative bacteria, and that gram negative bacteria attains counter stain saffronin and attain the pink or red color. This is basic staining. Alright, the simplest method of staining. Then moving toward the other type of microorganism. As we have discussed about bacteriology, now we will discuss about virology. As the name indicates, the study of viruses is called virology. The study of viruses is called virology. Here are different shapes of viruses. You can see here this is a basic structure of virus. This is another structure of virus, but it is enhanced structure. The center of this is taken out and then discussed. This is basically the structure having spikes on its surface. These are various morphological symmetries of virus, like this one is spring-like shape. This one is Icoashedral shape, just like a diamond, having different corners, different faces in the form of triangles. You can see here. Icosa word stands for 20, means this type of 20 triangles are present here in the icosahedral morphological shape. Then there is a complex structure of the virus. So these are the different shapes of viruses. different shapes of viruses. Then, explaining the virus is derived from a Latin word, as the word bacteria was derived from the Greek language, but the virus is a Latin word meaning venom, which is called as poisonous fluid, dangerous to human health. In the past, the virus is referred to as a point which is associated with disease and death. In past history, virus is termed as anything that caused disease and leading to death. That cause disease and lead to death. They are most widely used for the synthesis of vaccines. More of the vaccines are prepared from viruses. How vaccines are prepared? Obviously, we need to have a microorganism against that disease. If the disease is viral, then we have to use the virus which produce antibodies against that disease, and our body fight with it, and that resistance is attained in the form of vaccine. If we have a bacterial disease, then we have to use the vaccine made from bacteria, and that vaccine will produce antibodies in our body against that disease. They are insensitive to antibiotics. Viruses are insensitive to antibiotics. They are stable enough. Only antivirals are used for the treatment of disease or to inhibit the virus. Only antivirals are used. For example, if you are suffering from flu, then the virus which is used against flu is only used in that vaccine. Okay? You cannot say that the disease is viral and you will take simple antibiotic and that viral disease is treated. No. You need to take antivirals to treat a viral disease. For example, polio. You cannot treat polio with antibiotics. You need to take poliovirus vaccine. You will take a vaccine which is made from poliovirus. That is effective against that polio. We all had vaccines at the time of birth, polio vaccine, and that vaccine contained basically that virus which is used against polio. Basically a prophylaxis treatment. If the disease happened, then obviously polio is not recoverable, because that virus, we will study about different shapes of viruses. Depending upon the shapes, the viruses are divided according to the severity of disease. If polio happened, then the shape of virus that causes polio is such type which is very, very stable, which cannot be recovered, which cannot be reversed if the disease happened. Like polio cannot be reversed because the type of virus which causes polio is very resistant to any antiviral. So before happening the disease, we need to have a prophylaxis treatment like polio vaccine. So the virus will not enter in our body. If it enters, then we already have antibodies against that disease. So after the disease having a specific type of resistant virus, we cannot treat it. But before such type of viruses which are very resistant, then we have to first take prophylaxis before happening the disease. If we define virus, then they are ultramicroscopic. They are ultramicroscopic substances, non-cellular, having no cellular organelles, infectious, means they cause disease, entities. These are those substances which are basically non-cellular, disease causing, and ultramicroscopic, very, very small in size. For bacteria you study, they are microscopic, but viruses are ultramicroscopic. They are very, very too small in size and non-cellular and disease causing, which have either DNA or RNA. It contains DNA or RNA enclosed, packed in a protein coat. It has a covering made up of protein. It has a coat which is made up of protein, and that coat contained in it a DNA or maybe RNA. You can see here in the diagram, this is a coat, this is a cover of virus, and in it there is either a DNA, either RNA. Here is DNA and in this there is RNA. How we differentiate DNA or RNA? If the structure is double stranded, then it is DNA. If the structure is single stranded, it is RNA. And it reproduces inside a living host and cause diseases. It reproduces only inside the living host. We have discussed about it that viruses cannot grow artificially. They only grow if a living host is present. It causes disease such as hepatitis, influenza, smallpox, etc. It depends upon the host for growth. They utilize the host biosynthetic machinery for reproduction. Viruses are non-cellular. They do not have any biosynthetic machinery for reproduction. They cannot metabolize anything. They use the biosynthetic machinery of host cell. They use the biosynthetic machinery of host cell and increases their number inside that living host. And obviously if it reproduces inside a living host, then it will cause disease. How it is discovered? Edward Jenner in 1796, he prepared the vaccine for the disease smallpox. Against smallpox, Edward Jenner prepared the vaccine. Then Charles Chamberlain came and he called these infectious agents as filtrable viruses. They can be separated by the process of filtration. They can be separated. Then Ivanovsky in 1892, he said that the tobacco mosaic disease is studied and this disease is due to tobacco mosaic virus. TMV. Tobacco mosaic virus causes the disease called tobacco mosaic disease. You can see here, mosaic mean mottling, discoloration of leaves in the tobacco plant. Tobacco plant, the whole plant of tobacco leaves, complete leaves, all the leaves of the tobacco plant have a mosaic pattern, have a discoloration. This is the tobacco mosaic disease due to tobacco mosaic virus. Then W.M. Stanley. In 1935, he isolated tobacco mosaic virus in pure crystalline form. That tobacco mosaic virus was first time purely explained and isolated by W.M. Stanley. He said that the virus have a very simple chemical composition. The virus have a simple chemical composition. It only contain a nucleic acid and a protein. It only contain a nucleic acid, which is basically DNA or RNA, or protein. No other structure is present because it is non-cellular. As we have studied, the virus is a non-cellular structure. Then if it is non-cellular, then obviously it contains only some organelles, like it only contains DNA, it only contains RNA and protein. Then what about growth and reproduction? We have studied it that viruses are unable to grow in an artificial media. Like bacteria, we've grown the bacteria in a culture media, but we cannot grow viruses on that culture media because they do not have a metabolic machinery for production of energy and to prepare protein. They do not contain ribosomes to synthesize protein. It depends upon the host to carry out the life activities. The virus depends upon the host cell to carry out the life activities. Then they grow only inside the living host. a plant cell or animal cell, or maybe any other microorganism, or maybe human body. They may present in plant cell, they may present in animal cell, they may present in human body, they may present in another microorganisms, maybe in a bacteria, maybe in a fungi. They may present and cause disease and increase their number, grow inside that living host. For example, you have studied if virus present in plant, you can give an example of tobacco mosaic virus. For animal cell, you can say cowpox. For human, you can say thousands of diseases like polio, smallpox, and many others. Then, if microorganisms like if bacteria, then it may also cause disease in that. They reproduce by the process of replication. As we know, bacteria reproduce by process of binary fission, but the viruses reproduce by the process of replication. Then occurrence. Where they are present? They are found almost everywhere in every ecosystem. Every biological community contains viruses. It is the most abundant biological substance. The most abundant biological substance. It cannot be seen with naked eye, only be seen through electron microscope. The diameter is 20 to 300 nanometer, and nanometer stand for 10 power minus 9. As for bacteria, it is in mm, and m stand for micro, that is 10 power minus 6. Then, moving toward the structure, it consists of two parts, a central core and an outer core. The central core consists of nucleic acid, called as genome, which may be a DNA, may be an RNA, not both. The both cannot be present at one time. Either a DNA is present or either an RNA is present. Outer coat. It is a covering made up of protein which encloses genome. The outer cover, basically the outer envelope, which have genome in its center. It is called as capsid. The outer coat is also called as capsid. Here is a structure. The core contains nucleic acid and capsid contain protein coats surrounding the core. This one is the protein coat, and in the center there is a nucleic acid. Nucleic acid have DNA. Here you can see this is one is a coat, or this one is a capsid. In the center there is, this one is nucleic acid, and nucleic acid in the form of RNA, because it is single stranded structure. Alright. Morphological classification of viruses. On the basis of size and shape, the viruses are of following types. They may be helical, they may be complex, they may be icoashedral, they may be prolate. Four different shapes: helical shape, complex shape, icoashedral shape, and prolate shape. What is helical? They have a capsid made up of protein, and a central cavity having genome is present in circular fashion, forming a disc-like shape. Alright, they have a capsid made up of protein, the outer coat of protein, and central cavity having a genome is present in circular fashion, forming a disc-like shape. You can see different CDs or DVDs. If we place them one by one and make a whole rack, then we can see the center of that disc or CDs or DVDs is a hollow space. The disc of protein, also called capsomere. That different disc we arranged are called as capsomere. In viruses, same as that discs are called capsomeres, in the form of a hollow tube, making a room or area for nucleic acid, and the central hollow cavity have nucleic acid, either DNA or RNA. Okay, the center has a hollow cavity. Okay, I will show you the structure. Here is the helical structure. You can see here spring-like arrangement. These are basically called as capsomeres, and the center contain nucleic acid in it, either in the form of double structure called as DNA or in the form of RNA. Okay, then the virus are helical in shape, usually 15 to 90 nanometer wide and 300 to 500 nanometer in length, depending upon the size of genome. More the genome is, more the DNA is in length, the more will be the capsomeres, more will be the capsid increases in size. For example, the length of the helical shaped virus depends upon the length of the genome. Example is rabies virus, which causes a disease called rabies, happens by the occur due to the bite of rabid dog. TMV, tobacco mosaic virus. Then moving toward icosahedral, it is spherical somewhat. The shape refers to the presence of 20 faces in a geometry. It contains 20 faces. Icosahedron, it is a fusion of 20 equilateral triangles. 20 equilateral triangles forming a spherical shape, making a spherical shape that form a closed shell having identical protein subunits. But helical shape is not a closed form, but icosahedral is a closed shell. It contains nucleic acid in its closed shell covered with the protein, and that protein is in the form of 20 different triangles, 20 equilateral triangles. These 20 triangles are made up of proteins and form a shell, strong covering. The genetic material is enclosed in the capsid. The viruses in this shape are released in the environment when the cell dies, breakdown and lysis occurs. And the content means the DNA or RNA is released in the form of virion. The content in the form of virion, a mature virus, is released. For example, we can say in icosahedral shape morphology, the type of virus is a virion, which is a completely mature disease causing virus. Example is poliovirus, as I told you, a very resistant virus. Then rhinovirus, then adenovirus. Then there is a complex shape. These viruses are a combination of icosahedral and helical symmetry and complex outer wall, head to tail morphology. You can see here this one, two different shapes are combined. This one icosahedral shape having 20 faces, and this one is helical shape. These two are combined and form a complex shape. You can see here this morphology is of head to tail regions. This region is head region, this is a tail region. When head region is combined with tail region, then and this bacteria in this virus is joined with the host, then The virus uses its tail, this region. These spikes are used for attachment, and this nucleic acid is entered into that host from this tail region. You can see here head to tail morphology is unique to bacteriophages only. This type of complex virus having head to tail morphology is called as bacteriophage. As the name indicates, it could be a bacteria, but it is a virus. The word phage means virus. Bacterio means bacteria. Any virus that is infected that causes infection in bacteria, or we can say bacteria eating viruses. The virus which eat or engulf bacteria, bacteriophages. Alright, the virus which infect bacteria, and they are only the exemplified in complex shape of virus, having both combination of icosahedral and helical symmetry and head to tail morphology. Head is icosahedral shape and tail is helical shape. A bacteriophage uses its tail to attach with the host cell and release DNA into the host using tail as a channel. As a channel, it uses its tail region and transfer its genetic material in the form of DNA into that host, and that host is living, and that DNA replicates in that living host and increases in number and cause the disease. Example is pox virus. It is one of the largest virus which have complex structure. The most common type of pox virus is variola virus, causing a disease called smallpox. Small blisters formed on the skin. Influenza virus. These are basically complex viruses. Then some important definitions like bacteriophage, virus that infect and replicate inside the bacteria. And prions, a type of virus whose structure and function are uncleared. Whose structure and functions are not cleared, and may be infections. Example is mad cow disease, in which cows are unable to walk properly. They loses more weight and unable to walk. Second disease is the mysterious brain infection, in which destroyed brain and spinal cord. The disease destroyed brain and spinal cord. What are prions? Structurally complete, matured, an infectious virus. Then envelope. In bacteria, we have studied about a capsule, the outer covering, which produces resistance against the attack of drug. Here in viruses, there is a presence of envelope, which protected from various drug attack. In some viruses, the outer coat is made up of protein called as capsid and is further surrounded by a lipid bilayer membrane. As the outer coat we have studied that made up of protein is further surrounded by a lipid bilayer membrane that also have a protein. This lipid and protein structure is called envelope. Capsid contain protein, and along with this protein there is another layer of lipid. That lipid and protein layers are collectively called as envelope. It is derived from the host cell membrane. The host, either a plant, either animal, either humans, either bacteria, whatever it is, from that host cell membrane, the virus derived this lipid bilayer membrane and uses to form upon themselves, and against medicine it is used. It produces resistance in that virus. They play a vital role in viral infections undergoing the following steps. The first step is the attachment to the host cell. It attaches to the host cell membrane, then enter into the cell, then release the capsid content into the cell, and then that released content is then have a newly formed viral particle. That DNA covers around a new membrane, and that new membrane form a new viral particle. This is how a viral infection occurs. They are also responsible for the transfer of genetic material from one cell to the another cell. It also determines the stability character of the virus, such as it means it stabilizes the virus. It produces resistance in the virus against the disease. Like if virus is attacked with the chemical, then that envelope provide them resistance against that chemical. Virus will not die with that chemical. For example, as in today's situation, you can see that COVID-19 is at its peak, and that disaster is due to coronavirus. That virus is very resistant, very stable in nature. Resistance to the chemicals and physically inactivation. These two are the basic methods to make the virus unable to cause disease, but presence of envelope make them stable to these chemicals and these physical method of inactivation, like from using ultraviolet radiations we make the virus incapable of causing the disease. But some viruses, those having envelope, are resistant to these two methods. You can see here in diagram, this one is the nucleic acid, a viral genome, maybe DNA, maybe RNA. Then there are capsid around it made up of protein, and this protein is further layered with another lipid bilayer membrane. This lipid bilayer membrane provided resistance against chemicals, against physical method of inactivation. These are basic spikes which make it more resistant. This is a basic virus, enveloped virus we can say. As we have discussed morphological states, morphological classification of virus, the second one is on the basis of genome. Secondly, classify virus on the basis of genome. Genome, as we know, it is maybe a DNA or maybe a RNA. DNA is double stranded, RNA is single stranded. It is called Baltimore classification. On the basis of genome, the classification is termed as Baltimore classification after the name of the microbiologist who invented this, presented by David Baltimore in 1975. According to David Baltimore, the virus is divided into 7 subgroups based on genome, based on DNA and based on RNA. You can see here class 1, 2, 3, 4, 5, 6 and 7 different groups, abbreviated as DS DNA, DS small letters and DNA capital, SS small, then DS small, then positive SS, negative SS, then SS RNA RT, and DS DNA RT. You can see here basically this SS and this DS, these are basic abbreviations, and this positive and negative means this is a parallel strand, this is anti-parallel strand. For example, this one is a parent strand parallel against which from here another strand comes and join to form a DNA. That one is negative strand. The parent strand is positive strand, and negative means the anti-parallel strands which comes out to complete the structure is negative one. Then You can see here class 1, the method of replication. You can say dsDNA here, dsDNA pronounced as double-stranded DNA virus. Example is adenovirus, herpes virus and pox virus. On the basis of diseases they cause, the name is given. Then number 2, ssDNA virus. You can see here ssDNA, abbreviated as single-stranded DNA virus. Example is parvovirus. Then third one is dsRNA, double-stranded RNA. As RNA is single-stranded, but in case it causes disease, it is a double-stranded RNA virus. Example is reovirus. The word reo is very specific. For example, R stands for respiratory, E for entero, and O for orphan virus, having no specific origin. That's why the name reo is given. Respiratory enteric orphan virus. Number 4, you can see here class 4, it is positive sense RNA. You can say here number 4, single-stranded positive sense RNA virus. You can write it after abbreviation as single-stranded positive sense RNA virus, picornavirus. Basically in animal it causes disease, dogs basically. Then fifth one is single-stranded negative sense RNA virus, orthomyxovirus, rhabdovirus. Single-stranded negative sense RNA viruses. Then sixth one you can see here ssRNA-RT, very important. Positive sense, SS means single-stranded RNA viruses. The other name written here is RT, that is stand for reverse transcriptase, carry out the process of transcription. That's why reverse transcriptase, RT. You can see here that replicate through the DNA intermediate, reverse transcriptase. Then last one is double-stranded DNA viruses, that also replicate through a single DNA, single RNA intermediate. Here is DNA intermediate, here is RNA intermediate. You see here is DNA intermediate, here is RNA intermediate. This RT stands for reverse transcriptase, carry out transcription. Okay, and HAPA-DNA is an example. Now if we read it from here, it is double-stranded DNA, it is single-stranded DNA, it is double-stranded RNA. This one is positive sense single-stranded RNA, this one is negative sense single-stranded RNA, this one is single-stranded RNA transcriptase, reverse transcriptase. Then double-stranded DNA, reverse transcriptase. These are 7 major groups. Then on the basis of, you can see here, taxonomy, classify on the basis of taxonomy named as ICTV system. ICTV stands for International Committee on Taxonomy of Viruses. International Committee on Taxonomy of Viruses. I stand for international, T stands for committee, T stands for taxonomy, and V for viruses. Taxonomy basically is the characteristics, classification on the basis of characters. Viral classification starts at the level of order and continues as follows with the taxon suffix. Order is virals. Whatever the name is, we will add viral after that word. Family, whatever the word is, we will add varity. Then subfamily, we will add varinae at the end. Then genus, at the end we will add virus. For example, polio virus. Genus will be polio, or polio for polio, or for smallpox you can say pox, then pox virus. Species, the species name generally takes the form of disease of the virus. For example, if the virus is causing the disease of smallpox, then we will say smallpox virus species. Smallpox virus species. This is basic taxonomical classification of virus, ICT, International Committee on Taxonomy of Viruses. Then, on the basis of capsid symmetry, you have studied here the capsid may be helical in shape, the capsid may be cubical in shape, the capsid may be complex in shape, icoashedral in shape. Any shape of capsid may be present. On the basis of that shape, the virus is given its name. For example, helical virus, cubical virus, icoashedral virus, complex virus. Then on the basis of envelope, maybe the virus contain envelope, maybe the virus have a layer of lipid around it, but sometime if the virus do not have a layer around it, the virus is not very resistant, then it is non-enveloped. On the basis of diameter of virion, they may be of different genome length, different length of DNA, different length of RNA. On the basis of that, we will say that the virus is of different genome length, having different diameter. This is all for viruses. We'll have a slight discussion on it. First of all, we have studied about staining, that is basically types of staining, simple staining or differential staining, and special staining. These three types we discussed about gram staining in detail, in which there are three basic methods of staining. First of all, we have to prepare a smear, then we have to fix that smear, and then we have to stain that smear. For fixing the smear, we first take a slide, then add a drop of water, and then add a drop of inoculum over it, and then mix that water and inoculum and heat that to fix the smear. After fixing, perform gram staining. Using these steps, as a primary stain, use crystal violet dye, and as a negative stain, use saffronin. The first one is a primary stain, then we add gram iodine. The positive will take that iodine and fixes the crystal violet dye. Both fixes that dye, but after decolorizer treatment, the gram negative will decolorize. Then we have a counter stain to restain gram negative bacteria, and negative bacteria will take the color of pink or red. Hence we differentiate under microscope different colors, as we will see in this slide. Gram positive will shows like that, and then negative will appear like this. Then we studied about virology, which is basically the study of virus. Three basic different shapes: helical, icoashedral, and complex. The fourth one is prolate. Basically, if this icoashedral is present in bacteria, then it is called prolate shape. Simply, we haven't discussed here because it's a simple definition that if icoashedral shape of virus is present in the bacteria, then they are specifically called as prolate. They are ultra microscopic, they are non-cellular, they are infectious entities. They reproduce inside the living host only and cause diseases. Then it depends upon the host for growth. They utilizes the host biosynthetic machinery. Then the discovery led to the formation that the virus contain a simple structure, that is DNA or RNA in its center and a protein coat. In its growth, it grew by the process of replication in the living host. Because they do not have metabolic machinery, they uses the metabolic machinery for metabolism in the host. The occurrence is that they are found everywhere. Wherever the biological community is present, viruses are found. Its diameter is 20 to 300 nanometer, 10 power minus 3. and then in its structure it contains two parts, central core and outer coat. Sometimes the outer coat may also have an additional layer to prevent it from the attack of medicine. Helical complex, icosahedral and prolate are four different shapes. Helical means spring-like shape and the center contains that DNA or nucleic acid. If it is complex, then it contains icosahedral and helical in combination, having a head-to-tail morphology. Head is icosahedral and tail is helical. The tail region is used for attachment and DNA is released from that region and disease is caused. Then envelope, an additional layer surrounding the capsid to protect the virus from any attack. It provides stability to the virus like resistance to chemical and physical inactivation. Then classification of viruses on different bases, like on the basis of genome. Then on the basis of genome, it is Baltimore classification, having the virus in 7 different groups like double-stranded DNA, single-stranded DNA, double-stranded RNA, single-stranded RNA in positive sense, and single-stranded RNA in negative sense. Then single-stranded RNA with the process of replication, having RNA intermediate, and then forming reverse transcriptase. Enzyme is used. dsDNA, double-stranded DNA, reverse transcriptase basically. If we are using double-stranded DNA reverse transcriptase, then we have intermediate form of RNA. If we are using RNA with reverse transcriptase, then we have DNA intermediate. Both are reversed. Different examples you have seen here: adenoviruses, herpes viruses, pox viruses, parvoviruses, reoviruses, as I told you, respiratory enteroorphan virus, picornavirus, togavirus, orthomyxovirus, rhabdovirus, retrovirus, and HAPA-GNA virus. On taxonomy basis, they are of order, family, subfamily, genus, and species. We basically deal with these two, genus and species name. Like as in bacteria, we've studied Vibrio cholera. Alright. Typhoid, Salmonella typhi. In case of viruses, smallpox virus, or we can say variola virus. So we use basically the name of disease and then start with the genus name. Then on the basis of capsid symmetry, they may be helical, they may be cubical, they may be complex, depending upon what type of capsid, what type of protein symmetry it contains. And on the basis of envelope, they may contain envelope, they may not contain envelope. On the basis of diameter or virion, they may be on the basis of different genome length. As the length of central genome increases, the length of the virion increases. And it's different shapes, like helical shape, you can say a long cylindrical shape, center is hollow. As in center DNA is present, increase in length of DNA or RNA increases the length of that capsid. If that is in the form of cube, then increase in that cuboidal symmetry will increases or changes the shape as cuboidal from helical. If it is of icosahedral, then it contains 20 triangles making a shell, and in the center the material core is present, packed. Various diseases, as you know, because of virus that happens. Maybe you have learned, you have listened about smallpox disease, rabies disease, influenza disease, yellow fever disease, then you can say polio disease, you can say mumps, measles, then you can also say rubella disease, then hepatitis. All these are viral diseases. They uses virus either in active form, either in inactive form, any shape, any virus, any type, depending upon the disease. If the disease is causing symptoms, sometimes vaccine is entered in body and it only causes slight symptoms. That means the virus is inactivated or virus is slightly killed. Attenuated form of that virus is used. So our body gain resistance against that virus when vaccine is applied because it produces antibodies against that. Then if we talk about the virus which are live, like when vaccine entered in body, it may cause some severe symptoms, increases the temperature of the body. That is something like viruses in the live form. And when virus entered in body, our immune system start responding against it. Either the virus entered in live form or either it is entered in the form of vaccine. It caused the disease. Alright, then depending upon the type of disease, the type of virus depends. Either it is live, either it is killed, either it is attenuated. In our next lecture we will try to cover this type of topic also related to immunity. This is done for now. Thank you so much.